Good morning, everyone, and thank you for joining the HC Wainwright 2021 Global Life Sciences Conference. My name is Ananda Ghosh, and I'm an Equity Research Associate at HC Wainwright. While we are virtual this year, we are confident we are going to be able to provide value to you with over 425 companies presenting at this conference, as well as via our interactions through one-on-one meetings. HC Wainwright is a full-service investment bank dedicated to providing corporate finance, strategic advisory, and related services to public and private companies across multiple sectors and regions. We have a total of 18 publishing senior analysts and 493 companies covered across all sectors. Please visit hcwco.com for more information. From a logistics standpoint, please make sure to reference your virtual conference online portal that provides your individual links to your meetings and all presentations. Panels and all presentations are live and on demand online from March 9th to 10th. With that said, have a productive and enjoyable day. And I would like to welcome Olav Hellebo, who is CEO of ReNeuron Group, which specializes in development of novel stem cell therapies targeting areas of significant unmet or poorly met medical need. Olav?
Thank you very much, and it's my pleasure to be with you all here today. So yeah, I'd like to present ReNeuron to you. We are a publicly listed company in the UK. And my name is Olav Hellebo. I'll leave this picture with you of the top executives and board members, and maybe also highlight on the right-hand side here some of the eminent scientists we work with in the field of ophthalmology and subretinal surgery. We work with really the best in the field, and it's a pleasure to have the support of the people that you can see here. So we are a cell therapy company. We're based in the UK. We have a clinical team, including our chief medical officer in the US on the East Coast. We have all allogeneic platforms. Retinal and neural stem cells are the two lines that we work with for the moment. We have an orphan drug designation and fast track designation for our retinitis pigmentosa program, and we have phase 2A data that I will share with you today. Following that, we expect a single phase 3 study to take that program to market. We have a very interesting exosome program that I will share with you. And we did a capital raise before Christmas, so that has given us the potential to deliver all this data with the cash that we have on hand. So we have about an 18-month runway going forward with that. These were our platforms. The human retinal progenitor cells is a cell line we licensed in from Skeppen's Eye Research Institute at Harvard almost a decade ago. We use these to deliver them subretinally. We believe that's the right place to deliver the cells in order to enable engraftment and also get trophic effects in the right place. We've been able to cryopreserve these cells. That's not easy to do, and it's also very important to do. It allows global ship and store and use for clinical trials, but also commercially. And it's been great help for us in rolling this program out. We have a partner in China with Fosun Pharma, and other than that, we're unencumbered. The second platform is exosomes. So we've been working on exosomes for a long period of time. We harvest these exosomes from our CTX neural stem cells, which is a GMP line. So that gives us a really good starting point. We've proven that we can load them with different cargo. We've proven that they cross the blood-brain barrier. And we're working now with several pharma companies in collaborations to prove that not only can we cross the blood-brain barrier, but that whatever payload we are delivering, that is actually being delivered and is functional. So that data is coming over the next few months. Third platform is iPSCs. So our neural stem cell is immortalized. We have taken that backwards, so gone backwards to create iPSCs based on the CTX cell. That has meant that we've been able to preserve the immortalization. That has created a whole new iPSC platform with full freedom to operate in a field that's quite tricky normally from an IP point of view. And we obviously can create any cell based on that. And we're looking at some of the top targets, as you can imagine. I'm going to skip the pipeline because I think I kind of covered it already and in what I said, and we'll come back to some of it later. And I'd rather dive into our human retinal progenitor cell program and retinitis pigmentosa specifically. So RP is a good starting point for us. It's inherited degenerative eye disease. The incidence is makes it a large orphan disease, if that is a possible concept. The incidence is 80 to 100,000 patients in the US, probably 150,000 in Europe. Now, it's caused by gene mutations. However, it's not a great place for gene therapy because there are so many different genes involved. So every gene therapy will just treat a sliver of patients. Indeed, one of them is approved, which is Luxturna. It's approved for RPE65 gene deficiency, which is 1 or 2% of patients in this market. So the other 98% are not able to access any kind of therapy. The good news about what we're doing is that human retinal progenitor cells are treating the loss of vision, but it's not treating the genetic cause, and thus can be used for any of these patient groups. So the human retinal progenitor cells differentiate into functional photoreceptors in animal models. We see that there is an integration into the optic nerve that gives a broad range across different eye diseases, but we're focusing on degenerative diseases for the moment. I mentioned fast track designation, etc. We collaborate still with Skeptons, where we license this in, as I mentioned, and also UCL here in the UK. We spend a long time on the manufacturing process. Being able to produce these cells in the right quantity is important, and some breakthroughs in the manufacturing process enabled us to really get proper quantities and brought us to where we are today. The cryopreservation gives us 9-month shelf life for drug product. Obviously, that can be extended. And in terms of commercial potential, it's quite large, as RP is a substantial market in terms of number of patients. Luxturna has been launched. Pricing set by Luxturna kind of sets a ceiling price, which is a reasonable ceiling price, $850,000 per patient over 2 eyes is the US price for Luxturna, and the price is similar outside the US as well. So the surgical technique is subretinal surgery. It's exactly the same technique as commercially used by Luxturna, and we believe that is the right placement for the cells, both for the trophic reasons and also for cell differentiation. I skipped a page here, so let me go back. Phase 1 has been conducted. 12 patients were treated in the phase 1 study. We started with fresh cells, and fresh cells, that means a shelf life of hours rather than days or weeks or months. And then we introduced the cryopreserved formulation, and we took it into higher doses. And so that was done in phase 1. In phase 2, we treated 10 patients at the 1 million cell dose. And these were patients that had some visual potential, meaning they can read some letters on an eye chart. And I will share the data with you in a second. Mass Eye and Ear in Boston and Retinal Research Institute in Phoenix were the two sites for that these two studies. And so far, the efficacy data are very, very encouraging. We are excluding patients, or in this case, one patient who had no benefit from the cells due to the surgical procedure. You have to keep in mind that these patients have very poor retinas. They have diseased retinas. It's called retinitis pigmentosa because of the pigmentation that can impact the retina. So sometimes that happens in the patients that have received the cells. You can see a really great impact on vision in the treated eye. Typically in phase 2 studies in this area, you treat one eye and then you follow both. It's not a proper placebo, of course, and that will come later, but it gives us some indication. And you can see here 8 to 10 letter improvement across the different time points in the treated eye, and that's amazing. And obviously that is very much an approvable level of efficacy. Day 365, so 12 months, is most likely what the FDA is looking for in terms of efficacy endpoint. So if you look at the 7 patients that have reached that time point in the study, you can see that they all have had some improvement in the treated eye, but there's kind of varying degrees, some really profound efficacy, 10, 15, 20 letters, and some a little bit less, 2 to 6 letters. So we asked ourselves the question, what if we increase the dose? Can we get these guys on the left moving over to the right? Since we have no dose limiting toxicity. So that is actually what we decided to do, is to add another 9 subjects into this study where we double the dose, made some other adjustments to the protocol as well based on input from our advisors. We've added a couple of more centers. One is Professor McLaren's site in Oxford, UK. So we obviously welcome him into the study, and that's being open as we speak. Also Casey in Oregon, and we're planning a further center in Spain as well. So this is ongoing. We've treated so far, out of these 9 patients, we treated 3 of them. So there's 6 more patients to be included. And these are the next steps for the retinal platform. We obviously will continue to collect data from the first 10 patients, but these additional 9 patients as well, it's all about getting them now dosed and into the study and start picking up the data for those patients. COVID obviously doesn't help in all of this, but these patients are relatively otherwise healthy. So it's possible to do it, although it's a little bit more challenging than in normal times. We expect to have data later this year, so in Q3 we expect to have 3-month data that we'll then present into one of the large conferences later this year in ophthalmology. So following that, we expect then that the next step, assuming success, is to do a single further study, a phase 3 study. FDA has very good guidance on this. There's a guidance document, so we know more or less how that study should look like. It will be a randomized but not placebo-controlled. So that is, we'll randomize into 3 groups, high dose group, low dose group, and then the third group will be an observational cohort. So that's then patients who are not getting the good stuff, but are being followed over time. In terms of patient numbers, we give guidance of about 100 patients for a study like that. You don't need huge patient numbers to show a p-value in change in visual acuity. Just to share with you a little bit who else is in the market and how the landscape looks like. Now, there are 2 different approaches. One is cell therapy, as we're working with, and the other one is gene therapy, like Luxturna. in terms of cell therapy, that's obviously the only way to address all the patients. And there are only two companies that are pursuing that. That is ourselves and JSAT, which is a private company based in California. I've listed some of the gene therapy approaches here, and you can see that if you add up all the different gene therapy approaches, they probably add up to 20 or 30% of all patients if everything was successful. So there's still a huge, the majority of the market is still there. And in addition to that, I would say there's probably synergy between cell and gene therapy in this field, fixing the gene defect and then repairing the damage with cell therapy. Is a concept that can work very well. If we're able to create new photoreceptors with our therapy, these will be healthy photoreceptors, and there's no reason why they would disappear over time. So with that, I'll share with you quickly a little bit on our exosome before we go to a summary and Q&A session. So exosomes are becoming quite known now. There's a lot of attention to that field. These are tiny vesicles that are released by all cells in a body, and they communicate with other cells. And it's possible to hijack this as a liposomal delivery system. And that is exactly what we are working on. They can be used for diagnostics uses or as a therapy in itself, but it's the transport that we're pursuing. Our exosomes are very happy to cross the blood-brain barrier, maybe because they are originally from CNS, neural stem cell. We've shown that we can load them. We've shown consistent high yields. It's coming from a GMP process, so we started already at the GMP level. We have great analytics already established. And in addition to that, we can engineer them to target particular tissues. It doesn't have to be CNS. The way we're pursuing this is in research collaborations. So we've signed several large collaborations with large companies. It's about delivering their cargo, typically sRNAs or mRNAs. And the way this works is that they send us their cargo. We load that into our exosomes. We send it back to our partner, and they run in vitro and in vivo studies. Should we create a new product based on this, then we own the rights together. And in this case, most of that would mean that we would then license those rights back to our collaborator, because in this case, there are large companies that you would expect to want to take this forward. So we'll have data on that over the next few months. So we're obviously hoping to see a proof of concept in one of these collaborations. So in summary, I wanted to just show you quickly a little bit. There are some proper values in the field. When you look at what we're doing in retinitis pigmentosa, JSAT, our competitor here in the US, has signed a deal with Santen, a Japanese-based company, for non-US rights. Santen, JSAT's data was approximately 6 letters in the treated eye and about 3 letters or 2.5 letters in the non-treated eye, or in that case, the control eye. So they have shown some, I would say, modest efficacy, and that's exactly what we expect to them to be delivered in an intravitreal approach, because that is the big difference between ReNeuron and JSAT. JSAT puts the cells into the vitreous, which does allow for trophic effects, but not for any integration. And I would say that probably the trophic effect is in the wrong place, or not quite in the right place. So we're not surprised to see more modest efficacy in that trial than what we have seen so far. But there's still great commercial value even in that. So, that I think that's a credible deal, and hats off to Jay-Site and to Santen for taking that forward. And on the exosome field, you can see both Kodiak and Evoke signing important deals and collaborations. So there's clearly an area of a lot of interest. So the opportunity here is that we have excellent opportunity in terms of news flow over the next 6-12 months, mainly on the human retinal progenitor cells, but also in the exosome field, and through all of these collaborations that we have. So I think ReNeuron is able to show some great value in both of these fields. I didn't go into the iPSCs any further. Please go to our website to look at that. It is an earlier stage program on exosomes, but might have the potential is enormous. Thank you very much.